Okay. Hello, everyone. Welcome to the next Pages ECN webinar titled Radiocarbon Laboratories and What They Can Do for ECRs. We kindly welcome Irka from ETH Zurich. Irka is a geochronologist and she used radiocarbon dating in climate research, environmental studies, archaeology, cultural heritage, and forensics, as a lot of radiocarbon labs do. She gained her doctorate from ETH Zurich, where she's currently a lecturer at the geology department. She is also the president of the climate division of EGU and a member of the European Association of Archaeologists. And now I would like to give the word to Irka, and enjoy, everyone.
Okay. Thank you, Nick, for the introduction, and thank you for the invitation, Fernanda and Nick. That is quite an unusual title for me. C14 Laboratories and what they can do for early career researchers. I must say that I accept the title was given by organizers, and so I said, well, that's exciting, but of course I would not like to say what other laboratories should do for the ECRs, so maybe a short explanation. I will be talking about mainly my experience in a C14 lab as I started as a PhD student here 30 plus years ago. And I'm actually more, I think the path that I made, it's kind of path from the user to the radiocarbon laboratory person. I was, of course, lucky to be based here at the AMS facility in Zurich. So I will be speaking from the perspective of the radiocarbon lab based in an AMS lab. And my other affiliations are telling you also where I come from. I come from Poland. I studied in Krakow. I studied physics, and I have quite long collaboration with the university in Lublin, which is close to my hometown, which is Eastern Poland. So I would like to move to the next slide. Oh, sorry. It looks like that. Only this way. So that's the content of my talk. I want to talk about the potential of radiocarbon dating, and I would like to show the path that the samples are making in a lab without really giving a radiocarbon talk, because we want to talk about this, what the C14 labs are really doing and how you can, as an early career scientist, how your interest and activities can use the potential of radiocarbon labs. And then this is what I think is something, it's the core of my talk, how to find this information, despite of the fact that we are so connected and we have so much information. I think those tips, I hope, will be useful. I have this time is everything sentence from George Denton. Of course, you are a young scientist, so you maybe don't know this historical papers, but this is a paper on the glaciation of the Scandinavian ice shield. And I've seen George Denton speaking about timing of the glaciation, maybe in New Zealand. This sentence, time is everything, is what is a motto of my research. And I try, as a geochronologist, I try to convey this to all the users, collaborators. So to set up the background of what we are talking about, not going into details of the radiocarbon dating. You can see from this figure, my beloved figure with the Swiss cow here. You see this is made by a colleague, technician here in many years ago. Shows us the complexity of radiocarbon in the atmosphere, the atmospheric cosmogenic isotope produced in the atmosphere. We know that the basic is quite simple. We know the concentration, very, very low concentration, and we know where we can find and use the radiocarbon. So this is just to signal all those reservoirs where we can go and look at the C14 signal and study it. And then, of course, here we have this production of the radiocarbon. This is an interesting aspect for variability of the C14 in the atmosphere. So also a tool used quite intensely, quite new applications nowadays, looking at the really fine resolution of C14 variability, maybe due to cosmic solar activity. And then anthropogenic effects. This is something that we cannot go around it. It complicates our radiocarbon signal, but radiocarbon is again a useful tool in studying of the environmental effects of fossil fuel combustion, Suez effect, or using the bomb peak as a tracer for forensic, for example. To move here. Yes, this slide is just to highlight how wide range of this application is. And of course, I cannot see who is here, who is interested in what type of application, but you can see we go from malacological studies to soils. Here is a database that is recently published and under umbrella of pages. So later I will show also the where you can find the link to this. Applications to study paleoceanographic records, or here even groundwater radiocarbon is being used to date groundwater as well. So these are just the highlights and numbers. You see how many publications you can find with this, just the search of radiocarbon climate vegetation. Okay, this does not. This is a paper that colleagues of mine from radiocarbon facilities, mainly, most of colleagues here, we all put our heads together and we wrote this paper that is maybe not very easy to find, but it is written with people who are starting their career, they studies, PhD studies, or they starting new projects as postdocs. I marked the applications that we list here, suspecting for this audience that will be the box, and maybe wildlife conservation, biofuels as environmental studies. This paper, I will refer a couple times to it, gives you really overview of radiocarbon dating. So if you cannot find it, please contact me and I will share it with you. Let's see, it doesn't work, just return. Yes, this is again from this, it's a highlight, a preview or trailer of this paper, just to tell you what it includes. It describes the experimentation, how the radiocarbon is being done, how we deal with the limitations on the right side, how we reproduce our results. And in a blue box, you can see this variety of applications, which I already highlighted with just a few papers that I screenshot on a previous slide. And we have, as Namenta, this table. I'm actually most excited about this table, which had to go to supplementary informations, and I give you a screenshot of just one page. In this table, we put everything what we know about. You can see here carbon sources, the terrestrial applications starting here, but as the table continues, we have other archives and other proxies that we can date with radiocarbon. So again, if you're interested, please contact me and I will share this table with you, unless you can find it on the journal webpage. So this table is something that is helpful when you are dealing with your samples. And what does it mean dealing with your samples? Or what does it mean doing the radiocarbon analysis starting from sample preparation? This is what I call from the sample to the radiocarbon age path. And this panel shows you examples of, the tree is of course the simplest one. And then we have maybe macrofossils, and we have shells of foraminifera. We have beautiful charcoal at the end. All this sample material is being chosen. And now this is something what I already start here, my message to you. It can be chosen by radiocarbon lab, but it can also be chosen by you. And as I proceed, I will continuously say, go for this. Try to send material that you are certain that this is what you want to date, and you know your material the best. So all this can be, and currently I have many users, collaborators, and a lot of work that we are doing here in the lab is choosing the material that it should be used for radiocarbon dating for the accurate age of your proxy record. And then the next step is, of course, sample treatment, removing contamination, and the combustion or acidification of shells for the CO2. And the lower panel shows, of course, as I said, I am based in AMS facility. So we are talking about these very small samples. I can imagine this network is also interested in as small samples as possible. But there is also possibility to analyze larger samples with conventional methods. That is something that one can check with the laboratories as well. And the path that it is actually happening in the radiocarbon lab, and this is already the answer to the question in the title, what the AMS radiocarbon lab is doing for the user is this, is the graphitization. Of course, we can discuss about this possibility of preparing graphite and sending graphite powder to the lab, where it is pressed into this target. This is a cathode that is then organized for the measurement with standards, with blanks. Again, not going into details of the method. This is something that I invite you to read in a paper. And then this organized cassette goes to the accelerator. Here is an example of an accelerator that we are using here at Hengeberg at ETH. But of course, there are different types of accelerators also around the world. Yes, again, I'm referring to this paper with my colleagues where we summarized all those paths. And you can see here, this is an example of samples. Most of the samples will arrive in a lab like this, and then we unravel them. We'll look under binocular, and I will show you some of the pictures from material under binocular. And we go for the selection of the material, then later for treatment, and as I said, graphitization, AMS analysis. And as I have the results ready, I will calibrate and send them to the user, to the researcher or archaeologist. This is summarized in this figure, and I have actually this red box showing again, that's what the AMS lab has to do. It's not nothing that we can outsource to the users. However, I highlight this part of the choosing of the material and pre-preparation. That is a potential field that people, not being radiocarbon person, everybody is able to learn that. I'm really quite convinced about this. So yeah, I'm kind of repeating this figure again here with some insights how it can look like, the selection of material, washing of the sediment. Here I have the sediment core, and then it can be terrestrial sediment, or it can be marine sediment that I can separate terrestrial macrofossils of plants. Or I can pick out the foraminifera that later will be graphitized and measured with accelerator to provide us the C14 concentration, which can be here shown as a radiocarbon age, calibrated. And of course, we can here again, we can say those parts are not necessary activities that radiocarbon labs have to do. If you would like to take control of this part, this is at least in case of my lab or me, I very much welcome such samples, because those are clearly chosen by the users. Definitely, I think foraminifera separation, this work has been done. That happened long time ago, that users started to pick out foraminifera and send this to the labs, and the labs just graphitize or go directly for AMS analysis. And then in the last panel, we have the age depth model calibration. This is quite often, I provide this as to the users, to the researchers, but I realized that many of my collaborators, they are very skilled in using this calibration models, and something that is possible for everybody to learn. This is again something that it doesn't have to be done by the radiocarbon, or it can be done in collaboration. So I promised you some pictures from choosing the material. All of those are showing one problem. There is a route in each of the samples. The samples are different type, from peat to wood. This is sediment. This is beautiful charcoal, but with a little bit of a root that might penetrate inside. Here is another route. All this has to be detected. And this is something that radiocarbon labs do However, I use this opportunity to invite users of radiocarbon dating to do this work for themselves, just to be certain that you are sending the right material. I have a few more. This is actually quite nice wood in here, you can see. And here is a very nice organic matter in a sediment enclosed. However, if you look at, this is not ideal here, you can see here a mold that is formed in a wet sediment. Or if we continue here, you can see here very poorly visible, but this is dust that was sent together with a sample. This is cotton dust. That is modern carbon. That is a contamination of a sample. So all this is we are doing this in a lab. However, I again say that it can be part of the user activity. Next step, what I want to show is how the user can get involved in the separation of material. I have this picture of tools, quite simple tools, sieves. The mesh shown here allows us to remove roots. And also, if we sieve sediments, we might collect macrofossils that are in the sediment and pick those macrofossils out of it. You can see here how the setup works. This is the larger than 125 micrometer fraction, and the smaller one I collect in a beaker, and then I would centrifuge, and this collected sediment will be the total organic carbon that it undergoes the preparation. So yeah, macrofossils is the beautiful material that we all crave. I must say this is my thesis. This is the macrofossils separated from Soppensee in Switzerland. Beautiful birch seeds and flowers of lime tree, 7,000 years old maybe. So, you know, if you can find material like this, you are really lucky. Not always we are so lucky, but it is worth trying to search for such material. Here is material selected from peat. And this is actually work of technicians in our lab, that they were curious and they were picking out this material, and it worked nicely for the dating of the peat. So you can see how this separation of material can be also a kind of rewarding activity. The sample treatment and graphitization, some pictures from the lab with the sediment. And this is, again, I kind of focus on this treatment of total organic carbon with acid-base treatment in a warm cabinet. You can see how the reaction undergoes. We can see this coloring. Then is the freeze drying in a for the last step of weighing, and this is in a lower panel. Okay, here you can see we weigh the samples and burn them in elemental analyzer for those steps of graphitization and then measurements. So kind of repeating this previous figures. This is the just a summary of this step of separation. What does it bring us? And maybe you recognize that was in a picture with roots, the root here, how the student's work of a master student, how she separated the root and She also sieved this material through the 125 sieve, and the results, if I look at various fractions, I can say we see right away the root has minus radiocarbon age, or a fraction modern is higher than 1. That is telling us it's a modern root. It's an intruding material. This is what I'm trying to convey. Roots are such an intruding material into all kind of outcrops and pit sections. And then we have different fractions here, and we can see how different ages they do have. And without revealing, talking too much about this, I know this fine fraction is in agreement with the section that I have for this sediment, but also I can explain the younger ages of the not fine fraction and organic matter. They are younger because they might be contaminated by this root. We know that there is the root, or there are other roots as well, that they are contaminating all those ages. So not that is as much as I want to show you for the effects of choosing the material. And from my perspective, it's almost the most important part of radiocarbon dating is choosing nowadays, because the precision, accuracy of measurements is achieved quite high level in all the labs. Now we really have to be very careful in choosing the material and in preparation, avoiding the contamination, but also avoiding the contamination of the material per se, contamination with a wrong selection. How, now that I talk about this, how you can get involved, and I can imagine not everybody is sitting close to the radiocarbon lab. The question is, how do you find the radiocarbon lab? And this is perhaps not always straightforward question when you are based in geology department and not radiocarbon facility is close to you. But we have this help of the radiocarbon journal. We have, as a radiocarbon community, we have the radiocarbon journal that we published. Most of my papers are published in radiocarbon. The journal is based at Arizona University, and we have our editor. And nowadays, this is the Cambridge that is publishing it, but the webpage is still there. I learned from Nick and Fernanda that we can turn the complicated web pages addresses to this barcode, so maybe it works for you. The link will bring you to the radiocarbon journal with all those possible informations. And along, if we look here, is laboratories. This is what I want to talk about. If you go to this link, you will find a PDF that is listing the maps. Right now doesn't work here. Okay, so I made few screenshots there that show few of the radiocarbon labs. You can see here, starting from alphabetically. You can see already here, Argentina has radiocarbon labs, and then we continue. There is 18 pages listing various labs around the world. So each page may be 7-8 labs listed. And if I try again here, you can see here, it's not only AMS laboratories, because as I said, we are also able to do radiocarbon dating using conventional methods. You can see here Argentina has one of the laboratories might be AMS, but otherwise it's conventional. And I know this lab in Zagreb is a traditional conventional laboratory. However, nowadays they have also possibility to produce graphite targets, and they send those samples to their collaborative AMS lab. So there are different solutions on those lists. So you can just try to find the best collaborator for you. The list goes down to Uruguay. I see Uruguay has the conventional lab that is probably also available to students there. Then continue. If we continue on this list of labs, you can see here it ends with the lab code, and that's something that sometimes you can see in publications. The lab code is telling us where the radiocarbon analysis have been done. So this is something that you can also use to find the lab that you would like to send the samples. Again. Now to the question, which lab? We as a radiocarbon community, we all consider research that we're doing, it's comparable. I would say international radiocarbon intercomparison that is performed over the years. This paper by Marion Scott summarizes the gains that the community has from performing such intercomparison. This intercomparison tells us that radiocarbon labs are doing well. So I would say the advice is find the most excited collaborators labs, that they are interested in your research, in your project, and they supportive. And I think nowadays we can be sure sending samples is equally, how should I say? You don't have to look at the records of the labs. They are in this international radiocarbon intercomparison. There's another one coming soon. It was just presented at the conference here in Zurich, and I hope soon there will be publication of the newest results by Marion Scott and colleagues, that they organize, send the samples to the labs, and then collect the data. All this is quite an effort for radiocarbon labs, but we are all doing it just to see how good we are. So that's tells you also about the spirit of the radiocarbon labs. Now, talking about radiocarbon labs, I always say we have conventional and AMS labs, but of course we are all tending to move to AMS analysis because they are faster and they are on a small of carbon. So you can see here there's an upcoming paper. You can see that this summary shows you how the distribution of AMS facilities is geographically. clearly Europe is very densely populated, as well as Asia, Japan, and Korea, China, USA. We have need for maybe more facilities, not maybe, but certainly in Africa. Nevertheless, we do have facilities in South America as well as in Africa. So I am happy to show you the one of the facilities with the head of the facility is Corina Solis, and she sent me those slides. She's quite happy that I present this link to the Spanish link or English. You can find the contact is there, and Corina will be happy to support or discuss your research. Another lab I mentioned is the one in South Africa with the amazing AMS applications, and this is truly amazing applications on dating baobab trees. There's a lot of work done on those trees. However, the lab is not only doing the baobab trees, because that is quite an wide range. There is quite a wide range of applications, stable isotopes and radiocarbon dating back to 50,000 years. So with those two, I hope that I give you some insight to what labs are there to discover. I also want to show you what else you can learn from this radiocarbon webpage of our journal. There are links to the calibration programs and to the databases. And I made some screenshots from the, I would say, most popular. Calib is the program that has a large community using it, especially that we have here also the marine reservoir correction database by Reimer and Reimer. And we have also the Calib bomb. So I recommend this Calib.org webpage for users, as well as the OXCAL program that is from Oxford Group, from Chris Ramsey, with quite wide range of possibilities modeling, Bayesian modeling incorporated. This software is really user-friendly. You can also find a lot of support on this OXCAL Google group. So I really recommend that. Then the last, not yet last, but I mentioned the databases. This is really an amazing effort that not only radiocarbon community, but also archaeologists. And here is the PAGES program, collection of data, radiocarbon ages. It's put into databases that are accessible. You can find them on this web page. And then the last but not least is the email list that everybody can register to this email list. And in this way, you might keep in contact with what is happening in radiocarbon world. You can also send information to this email list, just register with it. And as you do it, you can get the news about upcoming conferences, job openings, proceedings. And this is an occasion also to show that there is an upcoming conference in Mexico City in less than a year. Again, Corina Solis is organizing it. So I invite people to have a look at this opportunity. Yes, this is my closing remarks, that I hope I showed you that radiocarbon dating is a useful tool. And hands-on experience and the work on own samples is really is something that I really want to recommend to everybody. Because you will know what type of material you send. You know the best your sample. It can speed up the analysis because the labs are busy and washing sediment is always an effort, needs space and time. Maybe it can be interesting financially. I don't want to speak for other labs. I'm definitely interested in people sending samples ready, and maybe that gives a reduction in a price. And then definitely radiocarbon laboratories are open to research questions and to discussion with users. We're really happy to exchange our information and also have a feedback from the collaborators or users. I don't really have any objections in sharing information. Right, this is my final slide, and I want to thank you for this interest in radiocarbon dating with a picture from the conference that we just had in Zurich in September. It was such a happy reunion after 2 years. Everybody was happy to be in Zurich, and I really think that everybody is welcome to this community. We had many students from who are using radiocarbon, not necessarily radiocarbon lab connected people, but using it in archaeology or climate research. Okay, thanks.
Thank you so much, Irka. That was you answered a lot of the questions that I already had. Let me first and foremost say that many of the links that you shared with us here, we can also.
I have to switch this off. I have to switch this off.
The YouTube page where we will put this recording, we can put extra text under that video, so we can put all the links that you wanted to share maybe also there. So once the recording is ready, we will put it in a newsletter and let everyone know. And so if you missed clicking on any link that Irka shared, you can probably also find it later on the Pages ECN YouTube channel. I see that there's at least one question in the chat from Ati. Let's see. Can you see the question from Ati, Irka?
Sorry, I have to switch.
Yeah. Mm-hmm.
Okay. Just a moment. No, new messages on. Okay, thanks for such information. Quick question, can you please explain how to carry out radiocarbon dating for fossil corals? Well, corals, it's carbonate, right? So basically what you need, you might contact the lab and plan the sampling. What we need is this few milligrams, 8 milligrams of coral will be about 1 milligram of carbon for the AMS analysis. And mostly people would just drill the sample and send the powder to the lab. You cannot really do more for this. There are possibilities to do sampling using laser ablation. This has been done in our laboratory. If you are interested in high resolution of the coral dating. But if you want to do conventional standard radiochronology, then just drilling of this coral is sufficient. Powder of 8 to 10 milligram of carbonate.
Yeah, has a follow-up question. They ask, but then during leaching process, they found there was no generated CO2.
During leaching, we found there was no generated CO2. Okay, just a moment. I really didn't switch off this one. I don't know why. I don't know why. What does it mean, leaching process? Is the person there to speak? Hello?
Let's see. Yeah, the person is there. Well, maybe we can try to turn on the microphone if they have a microphone. In the meantime, maybe you could also look at the before that. Above, there's a text in the chat from Ati, who asks a question about post-bomb radiocarbon ages.
AT.
Mm-hmm.
Okay, so I'm quick question about my own. What is a standard way of reporting post-bomb? For example, C14H of 710 for marine samples, and when calibrating with marine curve with local marines and H until the H564 beyond the standard. Oh. But are you sure that they are post-bomb? So that means that you still have that the marine correction is not really this sufficient. If you know that the post-bomb, the samples are post-bomb, they should be really negative ages, or fraction C14 has to be higher than 1. And if your correction is still not achieving this, that means that the correction is not sufficient. That means that the local marine reservoir age should be higher, right? To achieve that.
Okay. Yeah.
I think the questions like with the problems. You guys have my email. You can write me those questions to my email that I can answer in email.
Many of the questions are very specific.
Yeah, I work in lake sediments, and the most common problem in the radiocarbon date is the age reversal. Could you suggest any possible ways to avoid this? Okay, so that is maybe a good question to say. Yeah, right. I've seen many age reversals as well. Every lake sediment will be a specific setup, right? You might have age reversal through the slumps, for example, of sediment. Now, what does it mean, the age reversal? Are they too old ages or too young ages that you observe? Now, other question is, what type of material are you dating there? Are you dating macrofossils, or are you dating total organic carbon? That all these parameters have to be taken into account when considering the age depth model. So yeah, I cannot say what is the problem exactly in your case. All the issues, even the size of the samples that you are analyzing, it's a question that one has to look at all these factors. They might create the age reversals. The one that most obvious would be the slumps in lake sediment, for example, but also washing of organic matter from the catchment, that you collect the organic matter that is already old when it was deposited in it. So yeah, could be the reason as well. Yeah. Other questions?
Question by Asif Mohammadlone.
Yes.
Who asks, how good is it better, or how good is it to date carbon-rich sediments rather than the macrofossils in lake sediments?
Okay, yeah, that connects to the previous questions, actually, to the previous question, because, how certain you are that the bulk sediment will be the right material. And if you, okay, I really refer you to that paper I said at the beginning. There is in a figure, there is a study that we had in Zoppense with Merle Gierga. We looked at total organic carbon ages. We looked also at compound specific biomarkers. And in a way, we could see that as long as human activity didn't come to the catchment, that means 5,000 years ago, the lake was still recording carbon, which was contemporary to the sediment process. That means only when people came and disturbed the catchment, we started to see the washing in of the old carbon. So in a way, as long as we don't see any disturbance in the catchment, we can say maybe your total organic carbon is as good as macrofossils. That is, would be my answer. Yeah. So again, there is no general answer to those questions, unfortunately or fortunately, because it makes it interesting. Each site and each sample is specific. So radiocarbon is actually site sample specific. We can apply standard procedure to dating tree rings. We know tree rings will close the radiocarbon as the ring is formed, and that's fine. But lake sediments, even peat and marine sediments, all those carbon reservoirs are very, very complicated systems that they cannot be generalized. No. Yeah. I see Saraya. Someone else wants to ask the question. Ask to unmute. Can I unmute?
Hello, ma'am. Am I audible? I'm Shreya from India. I asked that question regarding that age reversal in lake sediments.
Okay.
So but regarding my samples, it is I had worked on the bulk sediments as there was no microfossils, and the problem with that old radiocarbon is also there. So, is there any way to just understand how we can avoid that thing, avoid the reversals by seeing any particular method or any particular analysis, if we can do it prior to doing the dating techniques, because it is usually we collect the cores and it is not possible for us to understand whether there has been a slumping or not just by seeing it. So that is my question.
Oh, it is a difficult. It is a difficult question, because if you cannot do macrofossils, if you cannot do, if you cannot do, maybe, people try to do pollen grains. They think of pollen grains. This is again very experimental. There is a lot of research already done, and it requires quite an effort to separate pollen and radiocarbon. But actually, I'm afraid I have no good answer to this. Tephra layers, some references that could help to fix the chronology.
Okay, ma'am. Thank you, ma'am.
Are there any other questions? This is your chance to ask them directly to Irka. And otherwise, I hope that we've shown that with any kind of questions, you should really feel free to contact your local or not local radiocarbon lab.
Exactly. Actually, that was I was trying to convey this message. I think that as I gave you this web page, and I realized that it requires maybe a better access to this information. We will be thinking about this. I'm already realizing this. But there is ways to find contact with local labs, which are really, I'm sure. I'm speaking now without asking my colleagues for the permission to promise you the help and responses of theirs. But I know from discussions what we have during conferences that we as labs, we learning from the problems, as the colleague just said about her lake. I mean, I say I have no answer right now, but maybe contact me and we can think about this. What's in your lake? When I look at your ages, how the depth profile is developing. And I know in India that map showed the New Delhi has a facility, and I know colleagues there. They working there. Seek help from them as well. So that is my main message to you. Try to find. Start the dialogue with the radiocarbon lab about what you want, even before you submit samples. It is worth to plan what you want to study. If it's one sample, then you kind of know. But if it's like a whole profile that you are trying to unravel a story, a study, have a proxy to date, then it's maybe good to talk to the radiocarbon specialist in planning this.
I think this is a good time to start recording, Fernanda. Thanks everyone for joining.